Hey everyone! So this video I'm going to explain the basics of action potential, how action potential spreads from your nerves to your muscles, and how this can cause your muscles to actually contract. So to understand how action potential works, the first thing you have to remember is that on the outside of the cell there are higher concentrations of sodium, and on the inside of the cell, there are higher concentrations of potassium. Also, the outside of the cell membrane is more positively charged than the inside of the cell membrane, which is more negatively charged, because even though potassium is positive, there are other components inside the cell membrane that are negatively charged, so the net charge of the inside of the cell membrane is negative. So the resting membrane potential is just the charge of the inside of the membrane when the cell is at rest. When sodium channels open, sodium channel is then free to diffuse from the area of high concentration outside of the cell to the area of low concentration on the inside of the cell. So this movement of sodium is what causes the membrane potential to go from being negative to more positive, which we refer to as depolarization. Now, repolarization is caused by the sodium channels close, then the potassium channels open. Potassium is then free to move from the area of high concentration inside of the cell to the area of low concentration on the outside of the cell. So this movement of positive potassium ions then causes repolarization, which is the membrane potential dropping back down to more negative. Now you'll notice that when repolarization occurs, the membrane potential becomes actually more negative than it was to start with, which is hyperpolarization, which is just caused by more potassium leaving than is necessary. And after that, after hyperpolarization, the resting membrane potential is restored by the sodium and potassium channels being closed, and the sodium-potassium pumping the sodium and potassium ions back to their original state. So this is what we call the action potential. It's just the change in membrane potential that's occurring because of the ions moving in and out of the cell. So why does this matter, and how does this cause anything to happen in your body? Here you see you have a myelinated nerve. The axon is covered with the myelin sheath. So in between those little myelin sheaths, there are voltage-gated sodium channels. Voltage-gated means that a change in the charge of the membrane will cause the channels to open. So when an action potential occurs in one part of the cell and the membrane potential in that part of the cell becomes more positive, it causes the sodium channels to open next door. So the action potential in one part causes an action potential to occur in the next part, and it continues along the axon tail, just like dominoes falling over. And so this happens all the way until the end of the axon, which is the axon terminal. So once it reaches the axon terminal, the voltage-gated calcium channels are opened, which allows calcium to enter the cell. Then inside the axon terminal, you have these little sacs of neurotransmitters. In the case of muscles, the neurotransmitter is acetylcholine. So these little sacs of acetylcholine are then released into the synaptic cleft, which is just the space between the nerve and the muscle. Once the acetylcholine is released into the synaptic cleft, it binds to the receptors on the sarcolemma. And if you remember, the sarcolemma is just like the sausage wrapper of the muscle. So it binds to those receptors on the sarcolemma, which open ion channels and create a change in membrane potential in that area, which we call the end plate potential. The end plate potential then opens up more voltage-gated sodium channels to open, which then spreads down the sarcolemma, just like the action potential spread down the neuron. So as the action potential spreads down the sarcolemma, it goes into these spaces called T-tubules, which have these little voltage-sensitive tubule proteins. So when the change in voltage happens near these tubule proteins, they change the shape, and they release calcium that has been stored inside a part of the muscle called the sarcoplasmic reticulum. Specifically next to the T-tubules, there's the terminal cisternae. So the change in shape happens, and then the calcium goes from the sarcoplasmic reticulum into the cytosol, into the cell. So once the calcium is released into the cell, it can then react with the actin and myosin. So you have to remember that muscle contraction happens because you have your little myofibrils, which were made up of your two proteins, your actin and your myosin, which when they react, slide over each other to become shorter and shorter. And this shortening, this becoming shorter and shorter of the individual actin and myosin, causes contraction of the muscle. So at rest, the active sites on actin are being blocked by tropomyosin. So this is preventing the actin and the myosin from connecting with each other. But when the calcium is released, it binds to part of the tropomyosin called the troponin. So the tropomyosin is the purple strand with the purple balls, and just the purple balls are the troponin. So the calcium binds to the tropomyosin, or the troponin, which causes a shape change to occur where now the troponin kind of rolls out of the way of the active sites on the actin that it's been blocking. And once the tropomyosin is out of the way, the myosin head can now reach the active sites on the actin filament. So once myosin can attach to the actin, that little myosin head kind of pulls and acts like a ratchet, and it pulls itself down the actin, which causes the shortening, which is where muscle contraction comes from. Whew. And that ratcheting, the attaching and detaching, is another process which I'm not going to explain in detail just a second. It's another process called Crossbridge Cycling. So I know that's a boatload of information. There's a lot going on there. My suggestion is to, in your textbook, find the pictures that explain what I've just explained, and now that you've heard me explain it, try to look at the diagrams and read through and see if you can make sense of it. A lot of people find it really helpful to create a list of the steps that are happening. You know, it shouldn't be more than half a page long. You don't need to, don't copy every single word from the textbook. Put it in your own words and try to make sense of it. All right, have a great day and good luck studying.